Okay, today we will talk about the blood. So when you first go to the doctor, usually he take the blood sample and put the blood in like the centrifuge, like this machine that you can see right here. And actually, we had this in the lab, so in the actual lab. So, but unfortunately, we don't, we just meet online. So when you put the sample of the blood inside this machine, it start to rotate and like about maybe a few hundred per minute to separate the blood component. Blood component is going to have the blood plasma and the blood cells. So if this is a tube, for example, and the blood sample, you finish the put the blood sample inside the centrifuge machine here, will actually rotate very fast. You're going to have some blood component at the bottom, which is heavier with the blood cells, and usually the rest will be like plasma, like the water of the blood. So the heavier cell, actually when you rotate it, the heavier cell stay in the bottom, red blood cells, white blood cells, etc., platelets, and the plasma stays in the top. Can everyone hear me clearly? Okay, great. So these are the blood component. Okay, will be the red blood cells. We call them erythrocytes. Erythro mean red. Cyte means cell, so this will be the red blood cell. The function will be to transport oxygen and carbon dioxide. The other component will be the white blood cell, which is leukocytes. Leuko mean white, cyte means cell. And this actually your immune system. This is the cells that defends your body against like bacteria, viruses, etc. So it actually destroys pathogenic microorganisms and parasites, and it removes worn cells and provide immunity. The white blood cells has actually two major components. The first one called granulocytes. Granulocytes mean they have in the cytoplasm. So if this is this shape, this is the white blood cell, for example. And cytoplasm, you're going to find they have these thick, like grain, looks like sand or grain inside the cytoplasm. Very specific one. Okay. The second component, the agranulocytes or non-granulocytes. There is no granulocytes, meaning the white blood cell does not have like a specific granules inside the cytoplasm. Okay. So the granulocytes mean again that this particular white blood cells has those specific stain or grains inside the cytoplasm. One of them will be the neutrophil. But these neutrophil phagocytes bacteria, the main function of this one will be phagocytes the bacteria. And actually these are the most abundant white blood cells in the blood. So when you take the blood sample, you're gonna find the neutrophil. They make about, probably we're gonna see it next slide, but I think if I'm not mistaken, between 40 to 70% of all the white blood cells. And usually this it's like when you stain, the reason that call it neutral from natural, right? Neutrophil. Phil mean loving, nature mean, like in the middle, so it's not acidic or alkaline or basophil, as we're going to see in the other two types. Eosinophil, which is acidic, sometimes they call it acidophil, and basophil, this is alkaline or basic. What that mean actually, this particular, like the eosinophil, the cytoplasm, you can stain this cell with acidic dye. With acidic dye, that's why this is looks like red inside. And the other one, the basophil or the basic, these cytoplasm usually they love the basic dye. We're going to look blue when you stain them. So any doctor, when they look at the sample from the blood, they look at the white blood cell. If they found that the cell, if they found that the cell like it's like blue from inside, for example, or stained blue, that means it's a basophil. And if it's actually stained with acidic or red, this will be the eosinophil. The other one has both the blue and the red inside the cytoplasm. So you're gonna find one red and one blue. Okay. You're gonna find both. Actually, you're gonna find this blue inside. That's why they call it neutrophil. Okay. And the basic one is supposed to be blue. Forget about this red dots. Okay, so when you stain them, it's supposed to be blue. This one inside is gonna be red, the eosinophil. Okay, great. And the non granulocytes, it actually like the granular cytoplasm. And first type would be the monocyte. It's actually phagocytized dead or dying cells and microorganisms. And the lymphocytes is the most abundant between both of them. Actually, this is I think it's make probably 15 to 40%, the lymphocytes or of the white blood cells. And it actually provides immunity. And when we study the lymph nodes, we're gonna find out a lot of details about the lymphocytes. And finally, the blood platelets or the thrombocytes. Okay, making thrombosis, make the blood cell to close the wound. So it's help control blood loss from injured blood vessels needed for blood clotting. Okay, so when you first go to the doctor again, you get the blood analysis. You take a little bit of the blood sample, put it inside the tube, and put the tube inside something called centrifuge to separate the blood component. So heavier cell stays in the bottom, and the rest will be the liquid of the blood. So the blood cells has two main types. One will be the red blood cells, erythrocytes, which transport oxygen and carbon dioxide. And the other one, white blood cell, called leukocyte white blood cells. And it's actually, this is your immune system destroyed pathogenic and microorganism. White blood cells has two main subtypes or main types. The first one called granulocytes. What that mean that when you look at the cell, you can stain the cell and find big granules or pieces inside, like looks like sand, okay, inside the cytoplasm. And you have three different actually dye. First one accept both dyes, the blue and the red, okay. And second one, the red will be acidic dye called eosinophil or acidophil. Third one will be the basophil. That's loving the basic dye or the alkaline dye. And this is actually releases heparin as anticoagulant and histamine. This is like it work as blood vessel dilator, like open up the blood vessels. The acidophil or the eosinophil. This is the image to show the blood component, which is like the erythrocytes, red blood cells, erythro red again, and leukocytes, white blood cells, and This is the specific one in the neutrophils, again, which is the most abundant one out of the white blood cells. And the blood platelets, that the one you see right here, this is for blood clotting. And if you don't have these platelets, so when you have even a small wound, you can bleed forever. So blood platelets is very important for blood clotting and stop bleeding. This is just a few subtypes of the neutrophil. As you can see, the neutrophil has like the multinucleated nucleus. So sometimes like this is bilobed, 3 lobes, sometimes it's 4 lobes, and it's like fine light purple granules. The nucleus single to 5 lobes, as you can see here. And the immature neutrophils called bands and have single C-shaped nucleus. This one, the immature one. And the mature neutrophil called sacs and have lobed nucleus. Just to show you a few difference and few shape of the neutrophil. This is the eosinophil or the acidophil again. So because of the acidic dye, see, take the red color here. It's actually coarse reddish granules, and the nucleus usually bilobed, has like 2 parts. Bilobed mean by 2, lobe 2 parts. So the nucleus always have 2 parts. Basophil, the basic again, blue, as you can see. It's coarse deep blue to almost black granules, and the nucleus often almost hidden by granules. So has a lot of granules to the extent that it actually covered the nucleus. Again, basic dye make this blue. Lymphocytes, this is agranulocytes again, the one does not have granules, or have granules but not specific, so you cannot actually stain them blue or red, okay? But agranule mean none, does not have granules. Slightly larger than the red blood cells, and thin rim of nearly clear cytoplasm. As you can see, like have a lot of, the nucleus almost cover the cytoplasm. See this part here, this little border, this is cytoplasm, this is cytoplasm, and it's larger, a little bit larger than the red blood cells. This is the entire nucleus covering most of the cytoplasm. So it's larger lymphocytes, hard to distinguish from monocytes. And monocytes, agranulocytes again, this mean does not have granules, none. A mean none again, does not have. Larger, the largest white blood cell, so this is considered the largest one, and usually this is actually the first one to run towards the wound or the inflammation. So when you have any kind of injury, this is the first one actually moved toward the sites. Cytoplasm nearly clear, as you can see, though you cannot stain blue or red, and nucleus round and kidney shape, oval or loop, as you can see. So okay, it's like almost bilobed. And finally, the platelets has several variations. It's actually the cell fragments, single to small clusters. As you can see, these little thing here called platelets or thrombocytes. If you don't have it in your blood, if you have even like injury, you're gonna bleed forever, and it's gonna be really a mess. Now the erythrocyte or the red blood cell, it actually lacks nucleus. There is no nucleus inside in the mature cell, of course, and the disc itself is looks like by concave disc, and thin centers appear almost hollow. See, as concave. See, it's almost hollow from the middle. Looks like a donut, right? And that's why they have this specific seat in the middle to carry the O2. So they have like the hemoglobin protein that actually keep the oxygen in place. The monocytes from 3 to 9%, and you can, if this percentage is high or elevated, that means the patient has malaria, tuberculosis, or fungal infections. Eosinophil or acidophil, the white blood cell that's stained by acidic dye, it's 1 to 3%. If you find this high, that means patient have allergic reaction, autoimmune disease, or parasitic worm. And the basophil, less than 1%. If you find this percentage high or elevated, that means patient has cancer, chickenpox, and hypothyroidism. So actually you can tell a lot about the health of the person by the blood analysis. That's why this is the first thing when you go to the doctor. They took like the blood pressure, the pulse, and they take blood sample to see the coagulation, agglutination. Okay, yeah, no problem. Now this is the blood. This is what we used to do at the lab, right? So you have two serum here. You have one anti-A, I'm sorry, and one anti-B. Okay. Of course, you take the blood sample and you put one. You have like this well here and the other one. Okay. You don't know what kind of blood here inside this well, right? So you're going to add anti-A serum and you're going to add anti-B in the other one. Okay. So what's going to happen here? You see this schedule here. So if you add anti-A and clumping happen or agglutination, you're going to find that thick liquid inside the sample. So it's going to be thick, stick with each other like this, called agglutination or clumping, okay? So if you add the anti-A to the first well and the anti-B to the second one. So if the clumping happen when you add the anti-A and no clumping happen when you add the anti-B, that's mean the blood is type A. You got it? So you have the two well here, one, second. You have blood sample inside, you don't know which one. You're gonna add anti-A here. You gotta add anti-B here. So if you find agglutination, that's mean clumping, thick, that is gonna stick with each other like this. And here nothing happened, still clear when you add the anti-B. That's mean this blood type A, because it reacts with the A, okay? And type A is almost 40% in the US, the average. Okay, now you add to this well anti-A and no clumping happen. And you add to this well anti-B and the clumping happen, then the blood type is B, and this almost 10%. You add anti-A to the first well, anti-B to the second one, clumping happen in both, that means this blood type is AB, and it's almost like 4%, the average in the United States. You add anti-A to the first well and anti-B to the second one, and no clumping in both, that means this is the O. Does not have the crown in the cell, does not react with the antibody, and it's 46%. This guy here, we call it universal donor, and this guy AB, we call it universal recipient, the lucky guy. Okay, so this is make it clear, or yeah, this is what you're gonna see in the lab. Oh, unfortunately, we're not in the actual lab, but it's okay. See, this is the anti-A in the left side, this one, anti-A, and this is anti-B, and this 2 well, right? So if you add the type A, you add anti-A and anti-B. Anti-B, no reaction. Anti-A, there is a reaction. That's mean this type A, or agglutination here, or clumping. See, become like thick. That's mean this blood type A. You add anti-A, nothing. It's clear. Anti-B, clumping. That's mean this blood type is B. Okay. So you add anti-A and anti-B, and there is clumping in both. So that's mean this blood type is AB. You add anti-A and anti-B, and there is no clumping here or here. That's mean in both well. That's mean this blood type O. Okay. There is actually third factor called rhesus monkey or RH blood typing. You add a specific like called anti-D. Okay, and this is was discovered by chance long time ago. It's mixture of the blood and the anti-D serum here. So if you add the anti-D and the clumping happen, that's mean it's rhesus monkey or RH positive. Okay, and there is no reaction happen, that's mean it's RH negative. Usually you put the blood sample and you heat it. This is called the RH or rhesus monkey typing box. You heat it up to 45%, 45 degrees Celsius or 115 degrees Fahrenheit. If the reaction happen here, when you add the anti-D, that's mean this blood type is RH positive. Okay, so you find sometimes you find the person has RH negative or positive. Okay, it depends on the reaction of the clumping. This actually what showed the reaction. So if anti-A serum is gonna look, you add it and there is clumping or coagulation, that's mean blood type A. Anti-B serum clear, that's mean this is B. And anti-D, the anti-D, this is for the rhesus monkey again, rhesus monkey factors. You add it, so if the coagulation happen, that's mean it's rhesus monkey positive, and this is a blood only on the control. So what? So if this is one patient and you added anti-A to the first well, the clumping happen. You add anti-B to the second well, no clumping happen, and you add anti-D and coagulation or clumping happen. Agglutination, I'm sorry. So what this blood type will be here in this image? Anyone know? Yes, Kriaki? Exactly, that will be A, because agglutination happened here, so it will be A. And since the anti-D reacted and the clumping happened, it would be A positive. This you can do